Stacy Bonos is an associate professor of perennial grass breeding at Rutgers University. She received her master's and PhD from Rutgers as well. Her research on switchgrass focuses on breeding and genetic improvement of switchgrass for biofuel production. The main objectives for her breeding program are selection for grasses with increased yield, disease resistance, lodging tolerance, germination rates, and cellulose to lignin ratios for improved performance on marginal land in the Northeastern US. So Dr. Bonos has experience with growing miscanthus and co-teaches an undergraduate course on biomass energy crops. She has received grants for her biofuel research from several agencies, including the USDA, the Northeast Sun Grant, Northeast SARE, DOE Feedstock Program, and the Energy Biosciences Institute. Her webinar today, in today's webinar, Stacy discusses breeding objectives important for switchgrass production in the Northeast. Switchgrass cultivars are very region specific, so it is important to develop cultivars within the region where growers will be using them. Breeding objectives may be similar in some regions and very different in others, and Stacy will guide us through that conversation today. So Stacy, go ahead and take it away.
Thank you, Sarah. Hopefully everyone can hear me okay. We'll just start by having a picture of me so you can see what I look like, and I'm 5 feet tall, so you can see the grass is at least 7 feet tall. In some cases it can get taller than that, but most of you have seen switchgrass before, but I just wanted to introduce myself a little bit. So we know that switchgrass is native to North America, so you can see the green indicated where switchgrass is adapted to. So basically everything east of the Rockies. And this is really interesting as a plant breeder because that's where the genetic diversity exists for improvement. There are two main ecotypes. Most of you are probably familiar with these, but for a plant breeder it's important to understand them and understand their limitations and their positive and negative attributes. So upland varieties are typically ecotypes recommended for northern latitudes. They are typically shorter, they have finer stems, they mature earlier, and are adapted to drier areas, whereas the lowland ecotypes are typically for the southern latitudes, and they're adapted to more wetter habitats. They're a little bit later maturing, they have thicker stems, and they're typically taller. In New Jersey and in the Northeast, and in parts of the Northeast and in the Mid-Atlantic, both ecotypes can be grown, and sometimes they differ in biomass yield, but sometimes they actually can yield almost equally. And this is kind of unique to the this region, because if you're in the northern states, you're typically just going to grow upland ecotypes. The lowlands don't do well in the winter. Whereas in the south, you're typically just going to be looking at lowlands. But here, we're sort of in the middle where you can grow both. And that's interesting for utilization of switchgrass here in this region. So this is just showing the great genetic diversity that we have within switchgrass. And you can see the upland on the right and the lowland on the left, and the upland is already flowering and it's shorter, and you can see the lowland is taller and actually hasn't even flowered yet. The seed heads are above the picture, so you can't, they've gotten cut off. But you can just see the differences that you can find within switchgrass. So this is useful as a plant breeder because we can select these types of plants and make improvements in them with future generations. This just is a little bit of an explanation about what I just talked about, having switchgrass, the upland and lowland ecotypes being able to be grown in this region. So this group did a study looking at multiple switchgrass biomass yield trials and found that the advantage of the lowlands relative to the upland disappears at about 42 degrees latitude. So if you're anywhere above that, you're typically just going to be growing upland varieties. And then if you're below about 35, that's where you're going to see just the lowlands. But again, here in the Northeast, we are right in the middle And so that's why you end up seeing both types be able to sort of yield equally well, depending on the year. Okay, so what are some of the breeding objectives that we're working towards here in the Northeast? One of the things when we first started doing this research about 8 years ago was that there was little to no information on cultivar performance in the Northeast. There was some work done at Penn State, but in terms of other regions throughout the country, throughout the Northeast region, there really wasn't a whole lot of information, and nothing in New Jersey. So, of course, biomass yield is an important characteristic that we're all interested in as plant breeders, and performance on marginal soils has really gained a lot of momentum recently. We also have a study looking at the cellulose, lignin content, hemicellulose in the plant, which I'll talk a little bit about. We have a germination study that I'll talk a little bit about, and end with some of the disease resistance breeding work that we're emphasizing in the new bio project. So that's just sort of an introduction of some of the topics I'll talk about today. We have multiple other projects, but we just don't have time to talk about all of them, so this is sort of a snapshot. When we first started looking at evaluating cultivars for performance in New Jersey specifically, we put out a study where we had 10 cultivars representing lowland and upland cultivars, and also ones selected from the Eastern US versus the Midwestern US, because one of the things that we know from Mike Hassler's work and others is that cultivars are very region specific. So one cultivar might do well in Wisconsin doesn't necessarily mean it's going to do well in New Jersey or Florida or other regions throughout the country. So we wanted to see what would do well here in New Jersey. So this is a picture of the trial. This was in 2008, so it was actually 2 years old at that point. And for this trial, we actually planted out individual genotypes instead of seeding it. So the data that we are presenting are on the individual plant basis. And we did that just because we wanted to make sure we got a good stand. And we know when you seed switchgrass that the stands aren't always the best. So we wanted to make sure we got a good stand. So we planted individual plants of each variety. And since we did that, we actually had to harvest the plants individually and get an average of the variety based on all the plants in the trial. So we tied up each plant with twine. You can see this here. This is my graduate student, Laura Cortez, who's just finishing her PhD. And then we come in with a hedge trimmer and chop down each plant and then take that back and weigh it. So it's a lot more tedious than harvesting on a seeded basis, but this is what you have to do if you wanted to look at the individual genotypes. So for breeding, we do a lot of this type of evaluation. And so when we look at the biomass yield, and you have to look at it on a relative basis because we can't compare it to a seeded stand. So we're looking at grams per plant here. So what we see are Cimarron and Timber are lowland ecotypes. Carthage is an upland ecotype, and you can see the relative response of the different cultivars. We found that Cimarron and Timber had the highest yields, and Carthage of the upland varieties had the highest yields. And then other upland varieties were not that great. Cave and Rock, one selection, NSU, is from Oklahoma State, and Brooklyn is another collection from Brooklyn, New York, and they were pretty low yielding in this trial. So interestingly, we find that even though Carthage was not the highest yielding, it was actually no different than Timber. So that's interesting for us. When we look at, not presenting all of the data that we took on that trial, but if we compare all the different measurements that we took, we find that Timber, which was an eastern lowland ecotype, somewhat similar to Bowmaster and Performer, selected out of some of those similar germplasm. It came from the Cape May Plant Materials Center, so it has some of that background in it. But we find that that is some of the tallest plants have the highest yields with the least winter injury. So for a lowland here in the Northeast, we want something that doesn't have a lot of winter injury. And then disease resistance was good, and so we are proposing that timber is a good ecotype for the Northeast or the Mid-Atlantic region. Cimarron, which I mentioned also had high yields, did have significant winter injury. So need to be a little bit careful about some of the southern selected lowland ecotypes. And then Carthage was one of the best performing upland varieties in the trial. So this was the first sort of in-depth analysis that we did on switchgrass grown in New Jersey. This particular project was done on prime farmland, so we just wanted to get a sense of what are the ecotypes we should be looking at for switchgrass production here in the Northeast. So we did find that both upland and lowland populations grow well with adequate biomass yields. And in subsequent studies that we've done, sometimes we get equal yields of upland and lowland varieties, and sometimes the lowlands are better and sometimes the uplands are better. So we find that it really depends on the rainfall. If you have a wet year, the lowlands are going to do better, and if you have a dry year, the uplands are going to do better. This is just showing some of the potential genotypes that we have. So if we look at that 900 grams per plant, which was the highest line that we got for some of those varieties in the previous study, we have ecotypes now that have or more than double the yield of the average of those cultivars. So we hope to continue the selection work and making improvements. So it does indicate that selection for increased biomass yield should be effective. So just to switch gears a little bit, talk about some of our research that we've been doing on marginal land. We know that the national strategy is to produce biofuels that won't compete for food production. So that's a given. But when we first started doing this research that I'm going to be talking about, I think there was one study that was done on marginal soils, and there was very little documenting the performance of switchgrass and other perennial grasses on marginal land. And as I mentioned already, we already know that switchgrass cultivars vary widely in their performance across different adaptation zones. So the question was, what was going to happen on marginal soils? And from a breeding standpoint, does that mean that I need to make my selections on a marginal soil to make improvements in a marginal soil? Or is the performance similar in both types of soils that I could do all my selection work on good soils, prime farmland, and then just utilize those in a marginal setting and have it be productive? Those are some of the questions that we had. We know that soil quality can influence the growth of plants, and this is actually a miscanthus project that we did and with the University of Illinois. And what you see is this is the University of Illinois clone in both pictures, planted on the exact same day. The picture on the left is the front of the field that has a good soil, and the picture on the right is the edge of the field where the soil gets pretty poor. So the plants on the good quality soil are already flowering and they're above Laura's head, whereas the ones on the right are basically at her waist and not flowering. So again, these were planted on the exact same day, and they were the same size plants when we planted the entire thing, and the plants are identical. So this is all strictly due to the soil effects. So we put together a trial that we called the U.S. Consortium for Breeding Perennial Grasses for Biofuel Production on Marginal Land. We had 6 cooperators representing several different regions. And each location, what we did was we planted paired fields. So we had a prime farmland site and a marginal site in each state. And we also had no nitrogen and what we considered a high nitrogen treatment, which was 100 pounds per acre. And then this is just a description of some of the marginal sites that we had. So in New Jersey, we had a shallow over shale situation. Rock Springs, our collaborator is Paul Adler, and this was in a fragipan soil. Wisconsin was a sandy, droughty soil. South Dakota was a shale, and then Ithaca had very poor drainage. And then I just included the mean temperatures and the annual precipitation rates as well. So you can see as you go further west, the precipitation goes down. So this is just a picture of what the marginal site looks like. This is in New Jersey, so what we find, it has maybe 4 or 5 inches of topsoil, and then you hit shale. So it's pretty bad. It's a class 4 site, so we tried to pick ones that were based on the USDA soil classification system. But the definition of marginal is up for debate, and we could spend the whole time talking about that, but we're not going to. But just we did pick sites that were below a class 3 for the USDA soil classification system. These were the cultivars we looked at, and we split those out based on whether they were northern upland, so selected from, North Dakota, South Dakota. So we have these varieties in here. We also had central uplands, which were, the Midwest region, and then we have southern lowlands, which are selected from the southern states, and then northern uplands, and the two there would be Canlow and Timber would be northern lowlands. So that's how we split out the cultivars. And then we established the plots, and each cooperator might have done this a little differently. We ended up doing it by hand because the plots were relatively small, and you can see the establishment on the prime site. And this is our establishment on the marginal site. You can see it's completely covered in weeds. But if we take a look at it, close your eyes for the first year and then come back, we did get some stand improvement in the marginal site here. And you can see the rows of plots in here, even though there is still some weeds in there. And then here's the prime site, and you can just see the big difference in yield that you can guess what's going to happen in the next few years. This is after 3 years, and so you can see the prime site on the left is doing quite well, and the marginal site is actually establishing much better, and the switchgrass is quite prevalent now in that field. But the yields are dramatically different that you'll see in a minute. We found when we analyzed all the data together that soil quality and nitrogen had the most significant effects on biomass yield, and it was more than the genotype. So the cultivar, even though it was somewhat significant, the soil quality and the nitrogen had more of an effect on the yield than the cultivar did. The yield ranged from about 7 megagrams per hectare in the marginal site to 8.8 in the prime site. And on the nitrogen, if you averaged it across the nitrogen, we find that it was 6.9 with no nitrogen and up to 9 with nitrogen. So the nitrogen, I think, had more of an effect than the soil quality did, which is somewhat, I guess, a little bit unexpected in that we assume nitrogen really doesn't have a huge effect on switchgrass. But Jeff Skousen at West Virginia found that when you're fertilizing on a marginal site, that the nitrogen has a lot more of an influence than you would expect on a prime site. This is just looking at the overall biomass yield across the locations, and what we see here is that there are big difference in the New Jersey prime and marginal. Here's the prime, here's the marginal. This was at Rock Springs, so the prime is, and then the marginal, Wisconsin, New York, and Pennsylvania actually planted an additional trial in Orners, and that one actually did really well for both, and then in South Dakota. But what you can see is that it's not, the prime is not always better in every single location. So that was something interesting that we found that I wasn't really expecting. But those are the results that we got. So, but interestingly, we do find that the yields in the Northeast are quite good, and they're better than what you would see in places like Wisconsin and South Dakota. So we found that biomass yield was approximately 1.5 megagrams higher in the prime soils than in the marginal. We would expect that. Nitrogen fertilizer increased biomass yield by almost 2 megagrams per hectare. The central upland cultivars established better than the southern lowland cultivars, and maybe this is just because of where we planted the trial. It was in a more northern regions, so that could be why. We found in New Jersey that the plants were 2 to 3 times taller on the prime land site than on the marginal site, and this was correlated to higher biomass yield. So, but when averaged across all the locations, we found a significant effect of prime versus marginal for every cultivar. So every cultivar had a significant difference between the prime and the marginal. But cultivars such as Shawnee, Blackwell, and Caven Rock actually had pretty decent yields in the marginal sites too. So these are promising in terms of what can we use on some of these marginal sites, and maybe a starting point for making some selections. Choose out of the ones that do well already. The next trial I'm going to talk about is a similar setup to what I mentioned before, but it's only in New Jersey. So we have our prime site, which is the it's called Adelphia here. I can't get my mouse to work. Okay. The Adelphia site, that's our prime site. Somerset, I mentioned to you already. And then we had a second marginal site, which was located in the Pine Barrens in Jackson, which is on a sand, class 5. And this is the trial we did. This was a clonal study, so we had 30 switchgrass clones, 5 replicates of each clone in 3 different locations, and we took evaluations for 2 years. And this was looking at the cellulose, lignin, and hemicellulose content across these different locations. And this picture was actually taken at the same time in all 3 locations. So you can see just the difference in growth between the prime site, which is flowering, very vigorously growing, lots of leaves, versus the sandy site in Jackson and the shale site in Somerset. Again, this will be all due to the soil. We calculated cellulose, lignin, and hemicellulose, and we followed the wet chemistry protocol that Dairy 1 uses. We also followed this up with NIR on all 900 samples that we ran, 450 per year. And that's how we calculated the cellulose, lignin, and hemicellulose content. We found that cellulose, that year and location accounted for the most variation. There was a significant effect of the clone interacting with those other factors. And we found that there weren't any clones that were stable across all the environments. But interestingly, for cellulose, we did find that if you were to pick the top 5 highest cellulose content clones in each of the locations, we have 3 genotypes that you would choose across the sites. So this Timber 3 was one of the top 5 in all 3. Carthage 1 was the top 5 in 2 of them, and Timber 2 was the in the top 5 for 2 of the regions, 2 of the locations. And we find that heritability for cellulose content was pretty high. So heritability is a measure of how much of the variation was due to the genetics and how much was due to the environment. If you have a 1, that means it's all genetic, and if you have a 0, that means it's all environmental. So if you have a 0.7, that's pretty good from a breeder standpoint in terms of it has a pretty strong genetic control, so we may be able to improve upon that cellulose content. So cellulose, I was pretty happy with the results on that. When we look at lignin, we still see also a significant clone by location and year interactions. The soil and the year also had a big effect on lignin too. And we had pretty low heritability. If you look at this number, this is 0.3. That is not good for a breeder. That means that there's a lot of environmental control, or influence, I should say, on lignin content in the plant, and makes it difficult to make improvements on. And we didn't see any stable clones, meaning a similar response across all the locations. And we didn't see the same thing that we saw with the cellulose, with the top 5 clones finding some consistent results. So I think for lignin, and we saw similar results for hemicellulose as well, I should mention. So I think we have a little bit more work to do in that area. So what can we say about the marginal land and the cellulose and lignin? The prime locations, when you do the heritability calculations for each location, the prime locations had higher heritabilities than the marginal ones. That's not surprising. The cellulose had relatively high heritability, so we should be able to make improvements in that. But hemicellulose and lignin, the heritabilities were quite low. Selection is not going to be as effective if we use the similar techniques that we normally use. So we need to come up with some either better ways of selection or some better ways to come up with making improvements. The next study I want to talk about is Switchgrass Establishment. We already know that switchgrass has very poor germination. It's slow to germinate. When it does germinate, the switchgrass plants put all their energy into the root growth and not so much into the above ground growth. So what happens is the weeds come in and take over if you don't have a very good stand. Weed control typically is necessary at establishment, but there aren't many herbicides labeled, so the main recommendation is to mow it. So this can be useful, but it would be better if we could improve the establishment of switchgrass. And we also don't recommend fertilizing because that just encourages weed growth. But it would help if you could irrigate. So since switchgrass has poor germination that results in slow and inconsistent establishment, we wanted to try to look at improving this trait. And if you look at the seed bags, here the germination percentage in here is really not good. 51, 31%, 21%. And the amount of dormancy in the seed can be pretty dramatic. So these are factors that definitely need to be addressed when making improvements in trying to improve germination in switchgrass. So we put together a study that we tried to say, our hypothesis was that if we select heavier seed, we should be able to make improvements and improve the germination, because that has been found that heavy seed has higher germination than light seed. So what we did was we actually had 3 populations. This is just representative of one population. And where's my pointer? Okay, there it is. We had our base population. We put the seed on a gravity deck, which separated the seed into heavy and light. And then we planted those seeds on a growth chamber, selected out the individual progeny, planted those into isolated crossing blocks in the field, harvested the seed, put that seed on the gravity deck again. For the light seed, selected the light seed again, and for the heavy seed, selected the heavy seed again. So we had 2 cycles of selection for heavy versus light seed, and then we also maintained an unsorted lot of seed. And then we did a large study at the end to compare all the different cycles for each of the populations. So we had a total of 18 different blocks. Unfortunately, what we found was that heavy seed did not improve the germination rate or the final germination percentage in switchgrass. And this is not what we were expecting. We were expecting heavy seed to improve germination rate. Interestingly, what we found was that just by going through the cycles of selection, regardless of the size of the seed, the germination rate and the total germination percentage did increase. So what that indicates is that we're probably selecting away from the dormancy that's present in the seed. So anything that germinated is what we went forward with. So that gives us at least some place to look for the future, and what can we try to do to address the dormancy issue in the seed, and maybe we can try to breed out some of the dormancy. It's not surprising that there's so much dormancy in the seed, especially when you consider sort of how far away some of the generations that we're working with are from nature. Dormancy is a mechanism built into the adaptation of the plant to be able to avoid adverse weather conditions before it germinates, and having the seed last a long time in the soil. So if we can maybe select against this dormancy, maybe we can select some varieties that have better germination. The last thing I wanted to talk about was some of the objectives that we're working on for the new bio project. And some of the things when we were talking about putting the project together, and what are some of the important things that really haven't been looked at yet, and they were diseases and insects. So as I've been working on this for the past 8 years or so, and we see a lot more diseases and insects sort of popping up. Rust has been pretty prevalent. Anthracnose disease is something that we found on our plants, and at first wasn't quite sure what they were, and I'll talk a little bit more about that in a few minutes. The switchgrass gall midge and the stem borer. The gall midge we've seen both in New Jersey and in New York, and the stem borer we've also seen in New Jersey, and as it has already been documented in the Midwest. So of course, spraying fungicides and insecticide is not an option for these low maintenance biofuel crops. So we need to come up with a tool, genetic mechanism, or some, through breeding, try to solve some of these issues. So again, anthracnose I want to spend a little time on, because that's one of the things that we're concentrating on. This is what it looks like. When we first started identifying this disease, we have a Choletotricum expert at Rutgers, and we sent some of the samples to her, and she had been doing an extensive study on Choletotricums across all different crops, corn, sorghum, others, turf grasses, and she realized that this anthracnose had never been described before. So we actually named the new species of the fungus, and you can see the name is Navitas. That's Latin for energy. So this is a Choletotricum species specific to switchgrass. It's been around for a long time. It just was never documented. So that's one of the things we're concentrating on for the new bio project, is trying to improve anthracnose resistance in switchgrass. We just put in a large breeding nursery, and this is one of our new bio scholars that we had this past summer help us in the trial. So we planted these, and we're going to be evaluating these for disease resistance. And we have some studies that we've done previously, looking at a range of cultivars for performance for anthracnose resistance, and we see a pretty big difference. A lot of the lowland ecotypes, Alamo, timber, here you can see, have pretty good resistance to anthracnose, and then some of the sort of less adapted upland varieties are pretty susceptible. Carthage, if you remember, was a pretty good yielding cultivar for the Northeast, actually has pretty good resistance. But what we want to do is try to get varieties that aren't going to have any breakdown in resistance and maintain good resistance over a long period of time. So that's our goal for the project. So what we're doing is evaluating our germplasm for anthracnose resistance. In addition to planting them in New Jersey, we also planted them on a reclaimed mineland in Pennsylvania. That's what this picture shows here. So we're going to be comparing the response in the two different environments. We have some data to indicate that there's a bit of a difference depending on the environment in which the plants are growing in terms of their response. And then we also have a mapping population that's segregating for anthracnose disease that we're also evaluating for disease resistance. And hopefully what we're trying to do is identify what type of inheritance it is. Is it just one gene? Is it multiple genes? Can we easily make improvements in this? Or are we going to have to sort of be a little more aggressive in our selection strategies? So I guess just to finish, I think switchgrass has potential to be a good biomass performance on marginal land, but I think we need to address the pest resistance, we need to improve germination, and we need to address some of the conversion either technologies or biology of the plant that affects the conversion technology. And I think all those things need to be considered. And this is actually another study where we're comparing switchgrass and miscanthus side by side. Again, it's a different study, but you can see the sort of differences between the two species. And I just wanted to end with this picture that we took on Friday. I think the snow is almost all melted now, but we've had a pretty rough winter in terms of trying to get some of the harvesting done. But this is switchgrass in the front, and that's miscanthus in the back. So there are some challenges still to work out with switchgrass. I wanted to thank all the funding sources that we have on the project, as well as all the collaborators and the graduate students and the staff that we have working here at Rutgers on the project. So I can open it up to questions, or if anyone wants to type a question in, be happy to answer them.
Yes, please type in your questions. I guess we'll kind of kick off the discussion. I just wanted to ask, so while breeding is a more sustainable and more long-term solution to managing some of those pest problems, I mean, what options exist right now for people who are growing existing cultivars or existing variants of switchgrass who have to deal with those kind of problems?
Yeah, I don't actually think there are any fungicides labeled for switchgrass. There may be, but I'm not familiar with any. Maybe somebody has some more experience with that. But I just think that we have to come up with a genetic alternative. If you already have the trial planted, that's kind of tough. I mean, the one benefit would be to plant, a blend of different grasses. That's sort of a way to if one variety is really susceptible to a disease and it's intermixed with some varieties that are more tolerant, you will reduce the effect of, you reduce the disease pressure on the one that is susceptible. So that could be a recommendation for something, a new trial planted. Or we're hopefully going to have a paper out pretty soon on the anthracnose resistance relative across the cultivars. So you can make your cultivar choices based on some of the disease data that we have before you plant. But something already planted is tough. And I know they've had some trouble in Tennessee. They have a big, huge planted area of alamo. And I know they had some rust problems in there. So it's a challenge. One of the questions we're trying to answer, also in the new bio project that I didn't show, is right now we want to see how does anthracnose influence biomass yield. Maybe it doesn't influence it at all. Maybe it just looks bad. And does that really matter? Just because it looks all yellow, does that mean that the biomass yield has been reduced because of the fungus infecting the plant? So that's something we're going to be looking at. We're going to be spraying half our plots with the fungicide and not controlling the disease in the other half to see what the effect is of the fungicide and the pathogen on biomass yield.
Great.
Okay, Dean.
Very interesting. There's a question from Damien. Did you see any morphology or maturity traits which correlated with cellulose content?
We are sort of working through that data right now. It doesn't seem to be um, it doesn't seem to be completely correlated. So I was assuming that the lowlands would have more lignin and the uplands maybe more cellulose, but it's not always the case. You maybe see a little bit of that trend, but um, I wish it was. I wish there were more correlations, because that would make it a lot easier to select. Like if you knew that early maturity was associated with more cellulose and less lignin, then you could just select based on that and not have to do the whole determine the cellulose content. But right now, I don't see anything that stands out right away that's correlated.
So he asks if there are specific types of marginal stresses which you think are more likely to lead to yield differences. So this gets back to a question that kind of plagues a lot of the work in this area, and that is the slippery idea of what exactly constitutes a definition for marginal land and how we deal with that in management.
Yeah. You know, one of our biggest problems on the marginal site was the weeds. And I think if we had gotten a better establishment on that, I think the yields may be a little higher. So if you could maybe try to somehow get a really good established plot, I think you're going to get higher yields that way. Some of the other stresses that I can think of. We're still looking at some of our soil quality data from all of our locations, and hopefully we'll have some more information on that when we get it all analyzed. But I think the water holding capacity of the soil may be an important factor. But in terms of other sort of characteristics of a marginal stress, I can't answer that fully, but those are some of my guesses. If anyone else has any comments on that too, we can discuss that.
Do you see a kind of pattern of classification of attributes associated with marginality guiding a sort of selection scheme for cultivars once there's a sort of larger suite of bred switchgrass available on the market?
Yeah, that's a good question. With the trial that we did with the marginal land study across all those sites, like I said, we're still putting all that data together because there is a huge amount of data on that. But they're all different soil, the reasons why they were marginal. So one was poor drainage, one was shale. So it's hard to say, you know, if we had done something where all of them were the same problem, then that would maybe be a little bit easier to answer. But since we had these marginal sites were marginal for different reasons, I would say that I didn't see sort of one variety stand out in every marginal site, and we would expect that just because you know, because of the diversity that we see within switchgrass. But the one thing that I did notice was some of the ecotypes that we were looking at from the Midwest actually had less of a variation between the marginal and the prime, whereas the ones that were selected, like in the Northeast, had a big variation between the soils. So I think there is a way to try to select some of these ecotypes that sort of have a more stable response across different environments than ones that really fluctuate depending on how much moisture is available or how much nitrogen is there. So.
Thank you. Mike points out that there are trade-offs with marginal sites, especially in terms of establishment costs. And it sounds like the example you gave where weed pressure was driving marginality would be a particularly good example of that.
Mm-hmm. Yeah, and sometimes establishing these marginal sites is difficult. You can't get in with the farm equipment. You have to use something else, and there's no way to irrigate it. So there's lots of challenges. So I think it's almost on a case-by-case basis. But what we're hoping is trying to get some answers in terms of how can we try to breed grasses specifically for marginal sites that are going to do well.
And then Mike follows up
Yes.
on not only lower yields but higher costs. Right.
That's right. And I think, if you look at the one marginal site we had, I think the yields were, equivalent to like 1 ton per acre. I mean, so low. Is that even worth establishing it? I mean, I think we even have to consider those kinds of things. The yields are pretty dramatically lower. And we want to maybe pick the marginal sites we use to be worthwhile. And I know Jeff Skousen's found that he can get pretty decent yields on some of his reclaimed minelands. So I think we need to sort of look at what are some of the marginal sites that we want to concentrate on.
Right, I'm glad you brought up minelands. That's what I had in mind as you began to talk about that. So there's a follow-up question about anthracnose resistance. So could you explain more about the genetic characteristic on that trait in your upcoming paper?
Okay, so for that initial paper that we're almost ready to get out there, is basically sort of a cultivar response, looking across multiple years at a number of cultivars, just to publish which varieties have good resistance and which ones are really susceptible. And then what we're hopefully trying to answer now is, what is the genetic control of it? Is it qualitative? Is it quantitative resistance? Because that will influence how well you can make selections. If you look back, if I can go back at that anthracnose data, the one interesting thing that I see is that you have sort of this cutoff here, where you have a whole bunch of low ones and then a whole bunch of really good ones. And so we have to look at this more closely, but maybe there's fewer genes than more genes. Maybe it's only a couple genes. And that's something that I don't know the answer to yet, but it's just interesting that it's not really a gradual line here. It's more like a bunch of susceptible ones and then a whole bunch of resistant ones.
So another question, is there a risk to eliminating dormancy? Are any of these plants that appear late in the planting year or even in year 2 coming from seed slowly releasing from dormancy?
That's a good question. So what you're saying is, if you eliminate dormancy, all your seed's going to germinate right away. And, maybe in some of these marginal sites, you get the seed that was dormant coming in and filling in and making the grass sort of, after year 3, have good establishment. That's definitely a good point. And something maybe that you need to consider. But I think maybe what the thing to do sort of early on is increase our seeding rates. Make our seeding rates even higher if you have a lot of dormancy. And I know we do, account for some of that, but it seems like it's not enough. I think we put out the average is like 10 pounds of pure live seed per acre, and you adjust for the amount of germination in there. But yeah, I think there is an adaptive advantage to have that dormancy in there, because it can maybe start coming in slowly after that initial establishment.
Yes, an interesting point. So I don't see, oh, I guess there is one more person typing in the chat pod, so we'll wait for that question. Oh, it's just a thanks. So that it seems like we're kind of coming to the end. If there are any other questions, please feel free to type them in. Stacy, do you have any final thoughts or comments?
No, just thanks for listening, and hopefully we're going to have some good results coming out of the new biobreeding project for switchgrass.
And we'll all look forward to it. Thank